Well, good afternoon. My clock says it's straight up 12, and that's when we start our I-29 Moo University Dairy webinars. Uh, Start right off asking folks to keep their mics muted and their video off. This is a fairly large group who will be attending today, and background noise just is a problem. We'd ask that you put questions in the chat box, and we'll keep them sorted, and we will address those at the end of the program. Today's program is the vaccine for avian influenza in dairy cattle. Our presenter is Dr. Alan Young. He founded Medgene Labs to assist in the translation of early stage technologies to the marketplace and develop new vaccine and diagnostic tools for human and veterinary medicine. Today, he will discuss how Medgene's platform is being applied towards the vaccine for avian influenza in dairy cattle. As I always have been known to say, Dr. Young, the podium is yours.
Thanks, Fred. I appreciate it. Thanks for everybody for joining us here today and taking some time out to hear about what we've been working on in terms of a vaccine for dairy cattle. First, a little bit about myself. I am a comparative immunologist by training, meaning that I got a PhD in immunology back in Toronto many years ago. And most of my career has been dedicated towards looking at the similarities and differences in how the immune system works and what its components are between species ranging from insects and fish all the way up to humans, but mostly focused on domestic animals. Um, I got my PhD in Toronto back in '94. After that, I had the opportunity to move to Switzerland for 5 years at a private research institute called the Basel Institute for Immunology, where I headed up the ruminant immune development lab back in the days when we were finding all the interesting stuff about how the antibodies are produced. When they closed that down, I moved to a short position at Harvard, just long enough to realize that I missed smaller Midwest type of lifestyle and took a job at South Dakota State University in 2001, where I have been on faculty ever since. Spent about the first 10 years working on mad cow disease with my colleagues at the USDA, and then pivoted to do vaccines once we figured out that feeding cows back to cows was pretty much a bad idea. Been working on that ever since. Formed Medgene in 2011 with my partner, Mark Lukey. And what I'm going to talk about today is the work that we've done at Medgene in terms of vaccine development for high-path avian influenza using our licensed platform that we currently have licensed for both swine and cattle. I am designing this a little bit like one of my lectures for my vet students here at SDSU. So the first half is going to be generically a little bit about the immune response and how we design vaccines and how they're targeted. I'm going to talk a little bit about avian flu and what we know about it, but I am not a virologist. I'm an immunologist. I focus on stopping it. And so then we'll talk about the vaccines a little bit. So first, the disclaimers. I am still a professor of veterinary and biomedical sciences at SDSU, where I serve as the assistant director of our professional program in vet med and teach immunology to vet students as well as undergraduates. But I'm also the founder and CTO of Medgene, where we have basically designed the work that I'm going to talk about here today. And the take-home message from that is you shouldn't assume that anything I say today represents either Medgene or SDSU. It's pretty much my opinion. I also serve as chair of the U.S. Animal Health Association Committee on Biologics and Biotechnology, which is basically the committee that focuses on the availability of biologics for the animal health industry. What I'm going to talk about today are what I know about dairy cattle vaccines for H5N1, so stuff that I've heard about H5, things that I know are going on in terms of the diagnostics. I'm going to talk a little bit about regulatory pathways. I am going to have a slide on what I know about what other companies are doing I was at a meeting in November when Zoetis presented their work on H5N1 vaccine, so I will mention that. And then I'm going to talk about our vaccine and what I know about where we're at there. So as I said, the outline for today, we're going to start off and talk in general about how vaccines are made and regulated for animals in the United States. I'm going to talk about the platform technology license platforms, or license guidelines, which is what we operate under, as well as how more traditional vaccines are made and what those kind of break down to in categories. I'm going to describe what a prescription platform vaccine product is because that's relevant to the discussion since that's what we operate under and that's how we design our vaccines. And then I'm going to finish off with where we're at with our H5N1 and walk you through some of the excitement that we've had over the last year working towards that goal. So, when we're designing vaccines, what we're really trying to do is we're trying to educate the immune system on how it needs to respond in the case of an infection. I'm going to focus on viruses today, but the general concept is conserved when we talk about bacteria and other things as well. So, what basically happens inside the body when an animal gets a virus infection is that virus in order to replicate, in order to cause disease, has to get inside a target cell. And once inside the target cell, it hijacks the normal processes that cells use in order to produce proteins and to metabolize and to live. The way it gets inside the cell is it attaches typically to some kind of a receptor or a molecule on the outside of the host cell. It uses that to get through the membrane and inside the cell. And then all of the normal processes from DNA coding for the RNA that ultimately leads to production of the protein is how it replicates itself because that protein ultimately assembles into new virus, which then is released from the cell and goes on to infect. So what we want to do when we're making a vaccine is we want to interrupt this process at some stage of its amplification. And so we could stop how that virus gets attached to the cell. We can stop how it replicates inside the cell. We can stop that cell from replicating. And that's really the target of how the immune system wants to take care of this virus infection. And our challenge when designing vaccines is to design a way to accomplish this without actually exposing the animal or the immune system to the pathogen in the first place. So the immune system basically has two arms that you learn about in vet school. The first is antibody-based immunity. Antibodies are these proteins that in effect are produced to stick to a target on the foreign pathogen. And by sticking to that target, in this case, this is another virus type, it can actually get in the way so that virus cannot get into the cell, or it can cause the virus to glop up together where it can't cause disease anymore. It basically is there to interfere with the infection process on the part of the virus. That's what you're probably mostly familiar with when you pull a blood sample and see whether you're still immune to measles or to tetanus or anything else. They're looking at these antibodies. The other arm is referred to as cellular immunity. For those of you who've gone through vet school, these are the T cells we're talking about. And while these two systems can't be completely separated, what cellular immune response does is it's more of a responsive side. So what it does is these T cells called cytotoxic T cells or killer T cells recognize when a cell is infected with virus. And what it does is it basically delivers a killing hit that kills this infected cell. So there's no clearance of the virus. What it does is it identifies what the cells are harboring the virus and kills them. And a lot of the symptoms that we get associated with viral infection are due to that. So what we target with our vaccines that I'm going to talk about today is largely this antibody-based system because it's more of a proactive system to prevent infection. So when I'm talking to the vet students again, I typically present it like this. Antibody-based immunity is proactive. It puts a guard, puts those antibodies outside the cell in order to stop the infective process before it begins. In contrast, cellular immunity basically throws up its hands and says, once you're infected, you're no good to us anymore, so we have to kill you. So it's more of a responsive. Both have clear roles in the immune system, but from a vaccine standpoint, if we can stop before it gets infected, that's what we want to target. So, with that in mind, how do we educate the immune system about these viruses? Well, there's basically three categories of vaccines that we routinely produce and that are routinely used in industry. The first, which you probably heard, are live or modified live viruses. So, what these are basically taking a live virus, typically of the same virus that you are trying to protect against, but you knock it back. You inhibit its ability to cause disease. You make it weaker. Or you can use a live virus that looks like the virus but doesn't cause disease. Smallpox, the very first vaccine that was ever developed, was one of these. We took a virus that infected cows, the vaccinia virus, that causes cowpox. But when we infect humans, it gives just enough infection to provoke immune response, but not enough to make you sick. And those antibodies that are produced in response to that infection are protective against smallpox. So you've never had smallpox, but you're protected against it. We do this today. A lot of the live viruses you're dealing with are what are called modified lives because they are reduced in their ability to cause infection. These are good because they're fairly easy to produce, assuming you can grow the virus. They are usually a one dose virus. You don't have to go back and give any kind of a boost. You get this strong cellular response that I was talking about, those T cells, the killer cells. However, in the field, we've seen some of these don't stay attenuated, so they can revert to virulence, we call it, so they can now cause disease. And if you've got any kind of immunosuppression, then in fact, these may be able to cause disease. And there's some other things. So if we don't want to inject a live virus, what about using a killed one? So we'll take all of this virus that's got all of its components, but we'll kill it so it can no longer cause infection. When we inject it, we then trust that all of these components that are part of the virus, the immune system will recognize them, produce an immune response, and protect against infection. These are very routinely used. They're the basis of autogenous vaccines that I'm sure many of you use regularly. They're safe because they're killed, so there's not a whole lot that can go wrong as far as causing any problems. They are easy to make, again, assuming you are able to grow and kill the target virus. In general, they're not as strong in the immune system. The immune system has more difficulty perceiving those as a threat. So that's why we add this stuff called adjuvant to it to try and make the immune system think there's something bad it's got to respond to. And that's what gives those side effects everybody knows about, the sore arm and everything else. And because it's got everything in there, the danger is you may want to produce the immune response against this surface protein, but your immune system may decide it's far more interesting to produce an immune response against one of the other proteins on the inside of the virus that is not going to protect from infection. So these diversionary responses. And then the last I'm going to talk about are these subunit vaccines, which is what I'm going to talk about today. These produce or isolate only that target protein that's responsible for infection and use that in order to expose the immune system. These are very safe. There's nothing alive in them. They're based on knowing which proteins you need to produce an immune response against. For many infectious diseases, this is known. For some, it's not. And so it can be a challenge. They are two-dose vaccines for the same reason that killed are. They're not really strong at provoking the immune system to be worried about anything. And you have to know what you're actually targeting. But with all of that in mind, it's a very targeted approach to generate vaccine antibodies to protect against infection. And so when we do this, to produce this subunit, say it's this protein here that is responsible for binding to the target cell and getting in, what we want is antibodies that will get in the way of this interaction and stop the virus from being able to get in the cell. So we can make, as a subunit, we can make that protein. And when you've heard about DNA and RNA vaccines, it's just a different way to make that protein. DNA vaccines, you get the DNA into the animal's host cells, and they produce this protein and release it to immunize the immune system. RNA vaccines, it's the same thing. The RNA gets into the cell and it produces proteins that are ultimately released. What I'm going to talk about today with Medgene is we go direct and we just make the protein in the lab and put that into a bottle, rather than relying on the host cells in order to produce the target that we're interested in. So I got a little bit of a video that we designed. As I said, what Medgene does is we produce the protein in a bioreactor in the lab. We harvest that protein, formulate that into a vaccine. That's what becomes the target that we introduce to the animal to provoke the immune response. What we operate under, and I'll talk about that a little bit more, are specific guidelines that allow us to use exactly the same system, excuse me, not symptom, system in order to produce a large variety of different proteins depending upon the infectious agent. So while I'm talking about H5 influenza here today, where we produce the H5 hemagglutinin protein, we also routinely produce and market vaccines for rotavirus, for influenza viruses of other subtypes, for influenza D, for coronaviruses, and we actually have vaccines against ticks and tick-borne diseases as well. So the way we do that is we first license and design our initial mechanism. And the analogy we use is like a Keurig coffee maker. So hopefully this will work. We'll see. Except, there we go.
Platform vaccine technology is like a pod beverage maker. Significant resources have gone into creating the maker so it can produce a wide variety of beverages as they're needed, quickly and economically. Just pick a pod, brew a beverage. It's fast and it's exactly what you want. Animal health vaccines made with platform technology are similar. Significant inputs of research, testing, and regulatory oversight prove the integral platform of a vaccine production technology is safe and replicable. Then, like a pod beverage maker, specific genetic information can be inserted into the platform to make a vaccine, and quickly, as the platform has already met regulatory requirements. Medgene's vaccine approaches leverage USDA-approved platform technology that is safely and easily adapted to multiple animal disease targets. The result? The development of vaccines and an understanding of how diseases move within species and geography within a fraction of the time of traditional vaccine approaches. Medgene. It's a new day for animal health care.
So that pretty much outlines the system. And so what Medgene has done is use these new guidelines to develop a pathway for producing animal health vaccines, under the new USDA guidelines that were brought forth in about 2018. So what that actually, I'm just going to get this back. There we go. So what this means is if we look at how vaccines are licensed for use in animals in the United States, this is a highly simplified overview of the characteristics. The USDA is mainly concerned about four primary characteristics: purity, potency, safety, and effectiveness. And what they mean by that is purity, that you know what you're making and that it's consistent and controlled, and it's going to contain the target agent that you need it to be. Potency, do you have a way to ensure that every time you make a dose of vaccine, it has the correct amount of that target protein that we were talking about in the vaccine? Safety is kind of a given, right? It's safe in the target animals. There's USDA mandated safety studies, and we'll talk about that one. And then effective, meaning that it does something to ameliorate the disease and it demonstrate that the vaccine itself has utility. For a full license, the kind that you go to Runnings or you go to your store and you buy a bottle of vaccine, these have gone through all four of these conditions. They have this so-called potency assay, meaning that you can pull a bottle off the shelf and guarantee the amount of antigen that is in there. They have assays that show you how to go into it with. They've obviously been demonstrated safety. And by effective, it means that there's an actual challenge study where vaccinated animals and non-vaccinated animals are both exposed to the infectious agent, and something is different between the vaccinated and the non-vaccinated animals. So in our studies, it can range anything from decreased symptoms to literally protection, such as our rabbit vaccine, our rabbit hemorrhagic disease, protection from death. So the vaccinates survive, the non-vaccinates do not. Below that is something called a conditionally licensed vaccine. A conditionally licensed vaccine is very similar to a fully licensed vaccine. It has purity, it has somewhat potency. You have to be able to guarantee that what's going into the vaccine is consistent and measurable, and you know how to do that. It has to have the exact same safety standards as a licensed product. Instead of a challenge study to demonstrate that it's effective, it has what's called a reasonable expectation of efficacy, meaning that there is some kind of a correlation that you can link to say that we expect this vaccine will work in the target animal. What I'm going to talk about with influenza today is the fact that we know in every species that we've got that if you have an antibody level on a specific assay called hemagglutination inhibition that is at least 1 to 40 dilution or greater, that you've got a protective vaccine. It's what we use for humans, it's what we use for animals. This is what we're doing now for our avian influenza vaccines for dairy cattle, because quite frankly, there is no challenge model, and I will come back to that. But that's the only real difference there. What we operate under largely are prescription product vaccines. And prescription basically says that we first had to get a fully licensed vaccine using what's called our platform, that pod beverage maker that we talked about in the last slide. In our case, we actually did it against influenza as our target vaccine. But then after demonstrating that, we are allowed to substitute the target protein that was in that fully licensed product that has gone through all the bells and whistles that takes 5 to 10 years and about $5 to $10 million to do. But now we can swap it out and replace it with others. And in exchange for that, we know that it's pure. We know that we can put the correct amount in using a formulation assay. We have done the mandated safety studies exactly the same as this for the target for that full platform license. But because we are swapping these out, we are not permitted to claim efficacy. So the vast majority of our products, efficacy is determined by the user in the field. We know what the target is because it's in the literature, but that's where it is. And then the last, which I'm sure you're familiar with, is autogenous. Autogenous is similar. It doesn't have the same level of purity. The master cells are not routinely tested and things like that. Potency, you have to have some kind of a formulation assay that is specific to the agent. There is no pre-licensing requirement for safety, and of course, there's no claims of efficacy. So with that background, what I'm going to talk about today are the approaches that we've used to develop flu vaccines for dairy cattle, a species that has not traditionally had a problem with influenza type A. D, sure. Type A, no. And the first message I would like you to take home is, despite the fact that last March came as somewhat of a surprise for everybody, influenza A in cattle is not a new event. We have known about this for a fairly long time. Going all the way back to the 1970s, we knew that you could take some strains of human influenza, inject cattle with it, and they would get infection. They wouldn't get very serious disease of any kind, but they would get infection. Studies back done in the late 90s demonstrated that, in fact, um, exposure to influenza type A, mostly swine flu, but also some human strains, was relatively common in cattle. And about 60% of the surveyed cattle had at least evidence of exposure to a level that would have generated antibodies. About 30% had strong antibody responses, implying that the cattle had been infected with this, and it continued. And furthermore, other studies demonstrated that the level of antibody, the amount of infection that had occurred in those animals, was correlated to a drop in milk production in dairy cattle. So all of this was out there, all of this was known, but it wasn't a disease of dairy cattle that anybody worried about until it became H5 influenza, another strain of influenza A, but unfortunately one with a little bit more political ramifications. So when we talk about H5N1, what we're talking about is the influenza virus, which has a large number of proteins. Out of those, the ones we always talk about are the hemagglutinin, which is the protein used to stick to the target cells and gain entry, and the neuraminidase down here, which is involved in that cellular entry process. And these hemagglutinins and neuraminases come in different flavors, hence H1N1, H3N2, and all of those sorts of things. And what we're talking about is a specific avian influenza strain known as H5N1. Inside, there are a number of other proteins, including this nucleoprotein, which I want you to remember later, as well as the PB2. When we look at all of the various isolates coming out of the dairy cattle, and we look specifically at the hemagglutinin, which is our target, there's over 3,000 now available in the databases. All of these are pretty much unchanged. They're almost identical to the original. There hasn't been a lot of variation in that hemagglutinin. Those variations you're hearing about are largely in this PB2, the polymerase protein, when they're talking about its potential to evolve towards humans. So, what do we need an H5 vaccine for dairy cattle to be able to do? Well, based on the California experience, I've had some communications with UC Davis and California. It's kind of been the epicenter of the big hit in the United States, so it provides us with some good information. First of all, the concern is that even though there is immunity, which I'll show in a bit, that can be found in convalescent animals that have been infected and that have recovered, replacement heifers, which have not been exposed, provide a continuous new source of naive animals for reinfection. So even if a herd has been hit and there's immunity there for a duration we're not sure about, if you're bringing new animals in, you're bringing in basically new food for the virus. So you have the potential for reinfection. We've seen abortions in non-lactating dairy cattle, and we don't know for certain that this is linked to H5, but it's intriguing. Natural immunity. We know there is natural immunity. I'll show you some evidence for that in a couple of slides. However, we don't know how long it lasts. We do know that we're now starting to see reinfection of herds that have previously had infection. I don't think it's as severe from what I've heard, but it's still there. It's still coming back. There is evidence for cross-infection of poultry facilities. That's fairly well known now. So the dairy cattle facilities that are adjacent or nearby poultry facilities, you can track the virus, saying that the poultry facilities likely acquired the virus from the adjacent dairy. We don't know exactly how, whether it's a biosecurity lapse, what's causing it, but we know that that virus has moved. As a result of using vaccine, we need to make sure that there is a way to continue animal movement in the face of vaccination. That vaccination on its own is not going to interfere with that, and in fact, may be able to enhance it. And we need to be able to tell the difference from a vaccinated versus an infected animal. If we can't tell the difference between the two, it's not going to help us with our trade or anything else. So, again, as I've said, Medgene is licensed in swine and cattle under prescription and platform guidelines. We've been licensed since summer 23. Our system is completely animal component free. Nothing derived from animals goes into it, so we don't have a contamination concern. It's a protein-based vaccine, and we use a killed insect virus that does not infect mammals as the way that we produce it in our cell cultures in the lab. The very well-known technology that's been around for quite a while. We started producing our vaccine actually as soon as we started seeing it appearing in wild mammals and carnivores. And we basically, back in 2022, added a new vaccine antigen construct, we call it, to our library. So we were prepared. When it hit, then it became a question of how we could best serve the industry by making sure that this vaccine was available. So some of the challenges to licensing this from a company perspective, first of all, it was a select agent, meaning that there is an extremely laborious process with Homeland Security to get permission to even touch the virus or work with it. And in addition to that is a biosafety level 3 isolation requirement. Now, with our system, we don't need the virus. All we need is an email with the sequence. But in terms of testing any vaccine, that means we can't get anywhere near that. There is still no relevant animal model for H5N1 infection of dairy cattle. We can infect dairy cattle intranasally or via aerosol, and they get sick for a few days and they clear it up and they don't get a mastitis. The only way we get the mastitis is to directly inject high levels of virus directly into the mammary gland. And we're pretty sure that's not what's happening in the wild. So we don't know how to mimic natural infection at this stage. And then, of course, there were initial trade associated restrictions on all animals used in the licensing studies. So I'm going to walk you through the regulatory stepwise. So remember, it was about the end of March that we actually saw that it was H5N1 that was the cause of the dairy problems. The first notice to come out of the USDA Center for Veterinary Biologics, which regulates vaccines and diagnostics for animals in the US, was an outline of requirement for diagnostics. We were looking for some way to be able to see this thing above and beyond PCR. There were several ELISAs out there. The IDEX NP ELISA, which was, I believe, licensed and in use in the US. And then there's a European company called ID Vet that has a couple of ELISAs against both the NP and the H5 that we started to use in our development. In May, the CVB asked for a survey of manufacturers on their capacity to produce H5 vaccine. Basically, how many of you guys out there can do it? There were a very large number of responses, but I think a large number of those came from very early startup companies or researchers. Because to my knowledge, for dairy cattle, there are only 2 biotechnology companies currently working. There might be 3, but there's 2 that I'm aware of. And that process of licensing typically takes about 5.5 years to get a license. And that's excluding getting your license to produce vaccine. So realistically, that's probably where we are. So initially, the restrictions were this was a select agent, it was BSL-3. If we were testing vaccines, they wanted even our killed vaccines that were routinely used in the field to be used in BL2 containment facilities, which massively increases the effort and the cost to test these in adult cattle. Any full challenge experiments had to be done in BSL-3 biocontainment. And I can tell you right now in the United States, there is not a BSL-3 containment facility sufficient to run a USDA regulated efficacy study using a direct challenge. None of them are big enough. We had no understanding of the natural route of infection, and all animals had to be disposed of following vaccination. So basically, from a vaccine company, we were at the stage where we could make the vaccine, but we couldn't actually do anything with it to test it as a startup company. I'll talk about that. Some of the larger companies were able to do things either offshore or at their own facilities, but that was a pretty strong limitation. Then in July, we started to get some different messaging. First of all, for the first time, the Center for Veterinary Biologics said that they would consider conditional licensure. So remember, I talked about full licensure as a challenge study. Conditional licensure means there's some kind of a correlation of immunity. And I talked about that 1 to 40 titer in what's called the hemagglutination inhibition assay. So CVB basically said they would consider licensing of a vaccine based on the antibody or serological response using this hemagglutination inhibition assay. And they would approve those protocols. For full licensure, they'll only do a vaccination challenge, which, as I've already told you, has to be in BSL-3, and nobody can figure out how to do it within the US anyways. And then basically, this little key down here is important to understand. Any licensed products only give you the license to sell if and only if the USDA decides that they are willing to allow US sale and distribution. And at the present time, that categorization is not available. So in other words, I'm going to tell you that we think that we're reasonably close to a conditional license for our vaccine. I think Zoetis may be as well. Neither of us will be given permission to sell that vaccine or distribute it. We'll just be holding it in reserve if that ever happens. So when we're talking about conditional, then our target is this line, which means basically some kind of a study to show that it has a correlate of immunity, that we get sufficient antibody to expect that it would be protective in the field. And we otherwise have to go through all of these other conditions. And so I'll talk about that a little bit coming up. Oh yeah, and then the last CVB notice, which really was the starting gate, is they noticed 2413 in August of 2024, when they then allowed us to use our killed vaccine products in field studies as opposed to under BSL-2 containment, with the proviso that there is a 2-year tracking requirement for any and all animals that have received the vaccine. And that also is limiting our capacity to do some of the field trials that would be of use. BSL-3 containment is still required for all challenge, and I talked about that. And the USDA has to give approval if you're going to slaughter them within 2 years. Finally, the efficacy assessment will be based on this hemagglutination inhibition assay that I'm going to talk about, which we have completed, and I'll present some data on. So it was really the end of August when it became feasible for companies to begin to develop, to go after the licensing pathway. We already had our vaccine available. If it were anything else, it would have been distributed back in March, but because they wanted conditional licensing, and that was required, that was the steps that we were going through. Again, no decision has been made yet to move forward and allow distribution. So let me prove this. I was at an international meeting in November last year, the International Veterinary Vaccine Network, where Zoetis presented some of their data. So that's all I'm talking about, is what I saw on the slide at the front of the room. So Zoetis has a recombinant influenza killed whole virus vaccine, which is an H5N2 that they developed for poultry during the 2013-14 outbreak and added that to the veterinary stockpile. Following the appearance of H5N1, Zoetis took this, modified it so it would have the updated H5, and began developing this for dogs and cats when we knew that mammals were being infected. Once it showed up in dairy cattle, they pivoted and did a serological study in dairy cattle with 9 animals. They saw these titers of antibody that would be sufficient for an expectation of efficacy in their vaccinated calves. And they have what's called this DIVA assay, meaning that they can tell the difference between a vaccinated and a non-vaccinated, because they use this N2 protein for the neuraminidase. The circulating virus uses N1, and so that would tell you the difference between the two. As of that talk, they reported that they were continuing to proceed towards licensing, meaning developing field safety, potency protocols, and things like that. And they do share some of the concerns that we have regarding the requirements in order to achieve licensing, including things like safe and pregnant claims for distribution. So we built our vaccine on the exact same platform as we use in our existing products for coronavirus, rotavirus, influenza D, papilloma, that coffee machine we showed you. If it were a common cattle disease like influenza D, the vaccine would already be available and released as a prescription product. But clearly, there are political ramifications to the use of H5. So we formulated our vaccine using this homologous protein to the current circulating strain known as 2344B. And we differentiate from natural infection. Unlike Zoetis, that's all that we have in our vaccine, that H5. So we actually can use that NP protein, which I talked about, the N1 protein, or other proteins to differentiate, since our vaccinated animals will only have antibodies against the H5. Also, our vaccine is undetectable by PCR. So any kind of PCR-based assays to monitor for live virus will not pick up our vaccine, even in the tube itself. So we're confident there. So I've talked about this hemagglutination inhibition assay at some length. Basically, what we figured out many, many, many years ago about influenza A is that if you take influenza A and you add it to red blood cells from the right species, this influenza will glop them all up. It will agglutinate these red blood cells. So if we put red blood cells alone into a little cup, a 96-well plate, and we let them sit for a bit, we look, what'll happen is those red cells will form down into this tight little pellet at the bottom. If we add virus that causes these red cells to agglutinate, then instead it forms this kind of a carpet or a mat in the bottom, and that's what's called hemagglutination. Now, if we add antibody to block the interaction of that virus with these red blood cells, in other words, we do exactly what I showed to block the virus from getting into the cell, then we block that hemagglutination, and we end up with this pellet again. So the titer that we're talking about, the antibody amount, is we know that if we can pull your serum and we can dilute it out 40-fold, 40 times what it would pulled, and it blocks, that you are protected from infection, basically. If, on the other hand, you only can dilute it out 20 times or 10 times, you're not protected. So vaccines target producing antibodies that can be diluted at least 1 to 40 and inhibit this red blood cell agglutination. So we found out very early on that, in fact, our target H5 that we produce in our system exactly responds the way the virus natively does. If we add the protein to red blood cells, we get agglutination. If we add antibody, we block it. So we can use that as an assay. And so we went back and we called up some people in Texas, and we got some of the convalescent serum from the Texas cattle. Remember, our target is a dilution of 1 to 40. We found that using our target antigen, we were able to detect immune responses in those cattle that had recovered from infection, where the average is about 1 in 230. So way above the level for infection. So we predict those animals are protected from infection. And in fact, they likely are. There's also a commercial ELISA based on that NP protein I talked about that shows infection. If you have a high enough amount of antibody, then you have seen that infection. And that, in fact, gave us a value reading of 0.68, which is in the positive range. So cattle that have been infected with H5 do mount an immune response. They do mount an immune response that is protective from reinfection, at least in the short term after infection. This was pulled within a couple of months of infection. So then we did another trial. We went back and gave our H5 influenza vaccine into cattle once we could. And this has actually been seen and approved for a claim of reasonable expectation of efficacy by the USDA. So with using our vaccine, we looked, oh, sorry, this is the ELISA. So we looked at 2 ELISAs, an ID vet ELISA, which is a little bit different. For this one, it's called an inhibition ELISA, a competitive ELISA. So we want the numbers to go down when we see protection. The placebo, we saw no change, so it didn't give us any response. The vaccinates gave us a strong response, which should be protective. So 14 days after a 2-dose vaccine, there's basically no detectable protein, meaning it works. Let me show you something even better. That hemagglutination inhibition titer that I talked about, the H5N1 vaccine, our target was 1 to 40. After the first dose, we had more than half of our animals had antibody levels more than 1 to 40. A few had them lower, so the average comes out to 1 to 30. 14 days, we're up to 1 to 280, so higher than the recovering convalescent cattle, so protective. And our placebos saw nothing. Based on this, the USDA has granted us a claim of reasonable expectation of efficacy towards our conditional license pathway. So the question is, how much antibody do we actually need in order to be protective? Well, there's a whole host of papers out there, but this one just recently came out from the the Vaccine Infectious Disease Organization in Saskatoon. And so here's the study they did. They took cows, and we know that you can't really get mammary infection by respiratory route. So they injected it directly into the mammary gland. And they found, we already knew that if you inject virus directly into one corridor of the mammary gland, it doesn't spread to the other corridors. It stays stuck in that particular corridor. So they did this. They came in and they injected one corridor, the rear hindquarters, with virus. And when they do that, they see the milk production drop and go down. And just as we know, it comes partially back, but it doesn't really come back. They then went back and they re-injected. Now in this one, they went into the forequarters. And of course, it does nothing to milk production. It does nothing here, but over here on this side, we're going to talk about the data. So they were able to get the H5N1 when they injected it into the hindquarters of the mammary gland to shut down milk production and to cause a transient increase in temperature. But when they went back, they saw no increase in temperature, so no evidence that there was any kind of a systemic disease at that stage when they injected it in the forequarters. This is the most interesting stuff, and I only want you to focus on this right side here. So when they looked at the local antibody in the milk coming out of those quarters, what they saw is after one challenge, they went up, and then the forequarters, the part they did not inject, they were seeing antibody titers of about 1 to 30, 1 to 40, so those minimal protective levels. And in fact, when they went out 10 days post-infection, they didn't see any change. So that tells us that having a concentration of antibody in milk of at least about 1 to 30, 1 to 40 protects against infection, even with a high dose of virus. So we know what the target is in the milk. So what does that actually mean? Well, we don't know for H5 exact. We haven't done the experiment to show what the correlation is between the antibody in the blood and the antibody in the milk. However, people have done it with other things, and in general, the correlation is extremely strong. On the right, somebody looked at bovine leukemia virus antibody in two cohorts, one where they had previous infection and one where they didn't. And they saw that the antibody levels pretty much overlapped between what you would see in the serum and what you'd see in the milk. Staph aureus, similarly, in this case particularly, they were looking at what happens if you inject vaccine in an oil adjuvant, and then whether you inject it in the udder or whether you inject it in the neck where we normally vaccinate. And they saw that the neutralization titer was pretty much the same regardless of the site of injection. So the take-home message from that is if we get high serum titers from an intramuscular or subcutaneous injection in the normal vaccination route, we would expect that if the titers are 1 to 40 or greater in the bloodstream, that they will be equally high in the milk and they will provide protection. So this one I've kind of gone through already, but basically one of the key aspects is to make sure that a vaccine will be differentiable from the virus infection. In fact, if we look at this, the approach that we have for Medgene, basically our vaccine does that. The NP protein assay shows when animals have been infected, but we will be negative for that. The PCR of the milk or of the animal itself will be positive if the animal has an active infection, negative if it's cleared, negative with our vaccine no matter what. And the H5 will be positive right across the board. So we can differentiate between either cleared or active vaccination and vaccinated animals. So again, this formulation is identical to what we've got. We've got over 500,000 doses already out in the field of these vaccines, not H5, but everything else. So we know its safety profile. We did not see any issues in the safety study, and we are just finishing off our 9 CFR USDA mandated safety study for our conditional license for calves. Little data, we did a milk drop study for those of you who are wondering, and the short answer is that compared to commercial vaccine where we did see a milk drop, in fact, with our either 1 or 2 doses of our vaccine formulation we're talking about here, we were basically indistinguishable from the negative control. So we didn't see any significant milk drop either. So the take-home message, and this is my last real slide, we are anticipating conditional licensing. We've done everything we have have to in order to get that. And basically, we think that this is exceeding the requirements based on our current license, but understand the confidence level that they want prior to any kind of vaccine support. This is exactly why Medgene developed its business model. Rapid response against emerging agents is what the prescription platform is designed to do. And it is the same as all of the other vaccines that we currently have. And we have that vaccine, which we're basically prepared to deploy should the need arise. We do support the USDA's point of view. We know there's a lot to go through before they decide to vaccinate for H5N1, but we want the industry to know that we are prepared should that come to pass. And with that, I appreciate your attention and am willing to take any questions. Happy to take any questions.
We would encourage folks who have questions to go ahead and type them in the chat box. We will go ahead and get to them as they come in.
Well,
starting right off with Jim asked, how often will we need to revaccinate?
that's an open question. We have to look and see what we need in terms of our what we have to do what's called a duration of immunity study. And the only way to do that is to vaccinate and go back and see how long the titers last. Typically, we do about once a year as our target for most of our vaccines. And given the titers that we're actually seeing, that's probable. Could be once every 6 months. It's not going to be less than 6 months, we would guess. We already know from our other vaccine products that we get duration of immunity that's good out to a year, so I'd be surprised. But I guess the correct answer is we won't know until we actually go in and do it. There we are.
I see another question that came in here, Dr. Young. There are at least 3 strains of avian bird flu in cattle now. Is this going to protect against all 3?
Yeah, as I tried to point out, when we talk about strains, what we're talking about is variation of the virus as a whole. The H5 is not varied. I'm aware of 2 strains. I'm not sure of the third one. I know the D1 and 1.2, and that one has been analyzed by researchers. And cross protection actually does occur. There's a little bit of a drop in protection, but we're still well within the margin for protection there. So we see no issues with that one. Now, that being said, one of the advantages of our platform is we can very rapidly add new ones and change. That's what we're built to do. So if something happens where we need a new one, we can be up and running within a matter of weeks.
Okay, can you speculate on how long it will be until USDA approves these vaccines?
Sorry, I'm laughing. I can speculate, but I don't know. As far as we're aware, we should have completed our part by the summer of this year. I don't know where any of the other companies are at, and I don't know how long it will take the USDA to on their end. There may be some additional steps that we are unaware of. I'm not even sure where that is. Now, the other thing I would say is from an approval standpoint, approval doesn't mean distribution.
And just a follow-up comment to the 3 strains, they commented the one that's everywhere, the one in Nevada and one in Arizona.
Yeah.
And then another question that came in, how quickly would it protect a cow? Do you see it being used when the virus has been ID'd in an area or just routine?
So we get this question a lot. That the way that I don't know how it would be used, but first of all, in terms of protection, I'm going to give an example or a story from something we had. We came up with a vaccine against a rabbit hemorrhagic disease, which is sounds as a really ugly disease. The rabbits die, they bleed out. It's not good. When we first started introducing it, we had a large number of clients who would call us the day that the virus was identified in their group of rabbits and ask if they could get the vaccine. Ours is a 2-dose vaccine to provide protection, means you're looking at about 28 days following vaccination before you've got protection. So the way that the USDA Red Book outlines it as a response to these would probably be the vaccine would be used in conjunction with eradication efforts. So it would be used in areas in terms of ring vaccination or to try to control and tamp down regionally breaks. But again, that's entirely up to the USDA. We're prepared for it, but all vaccines have to be used before you start identifying it in the herd.
And then there was a question on the vaccine and the platform technology. Is there any vaccines that use this platform technology on the market already?
Well, as I said, we've got 500,000 already out. We use this routinely. We're the only prescription platform licensed technology for use in cattle. There are other ones out there. There's an RNA from Merck that has been licensed for many years that's used widely in swine. The basis of our vaccine, the way we use it, is different than the existing licensed products. But yes, animal health vaccines have used a baculovirus production system for many, many years. What's different is that because the way we apply the system, we have a greater degree of control from the initiation of the actual production process. I mentioned the fact that we get an email with sequences. That's where our government regulated outline of production begins. Whereas for most vaccine companies, it starts significantly further down the line.
Okay, and Aaron has a question that's gets into the weeds pretty quick. So if you can see it, you maybe can read it and answer it. If I have to read it, I may
Let's
stumble
see.
through it.
I might have to ask you because for some reason my
Okay,
one of my windows is MIA. So,
okay. Question on antibiotic levels in negative cows, the NP and ELISA. The values were higher in convalescent cows. However, the NP and ELISA, the IDEX values should go down with infection. This was of interest during IDEX and ELISA verification for cattle serum, as the cutoff threshold needed to be raised. Was a different NP and ELISA used for those assays?
I have to check on that. It isn't, I think it was, yes. Because I know that one that first slide that I presented, the NP ELISA, um, that one was a direct. So yes.
Okay. Aaron, if you need more clarification, go ahead and add it. But man, she did. Also regarding the DIVA for Medgene's vaccine, since it's not completely clear how long NP ELISA detection persists post-infection, do you foresee reliance on the NP and ELISA as a DIVA as problematic, like for vaccinating post-convalescent cattle?
So all of those duration studies are going to require physical time in order to define it. Ultimately, any decision about what the DIVA, ELISA, or DIVA strategy may be on the part of the USDA it is up to the USDA. We presented that the compatibility for the DIVA access existed with some of the currently available assays or assays under consideration. It may very well be that their choice is to use something else. They may want to use the neuraminidase. They may want to use a matrix protein. They may want to rely on PCR. I really don't know, but that's up to them. So um, ultimately, all of those DOI studies that you're asking about are going to require time, and so that's the balance until we get out there. The only way to tell if something's got a 6 or a 12-month duration is to go back and check it in 6 or 12 months.
And I'm picking apart of a question. Can a vaccinated animal spread infection?
So the short answer is we don't know. That has to do, so the goal of any vaccine, okay, we hear a lot of talk about things like sterilizing immunity, which doesn't really exist in animal vaccinology to speak of. Um, the the difficulty to answer that one lies in the fact that we don't know how it's spread right now. So we do know because we've used this vaccine in a couple of other species that we very effectively shut down transmission, which is the question. So an animal that gets infected post-vaccination doesn't spread disease. However, in cattle at this stage, we don't really know. The goal from vaccination ultimately would be to shut down transmission. And that's the case for anybody. And I think anybody would say that. So, but I can't claim that because we haven't measured that in cattle because we don't know how to measure it in cattle just yet.
I know we have several marketers and exporters on the program with us. So I'd like you to reiterate, an export market buyer can tell the difference between disease and vaccination.
Yeah.
And have they accepted that?
I do not sit within the USDA's trade negotiating team.
Okay.
So,
Have
so
they
yeah.
done it in any other of the products coming from vaccinated animals, meat animals, or?
I know they used a diva strategy internally in Europe a few years ago. As far as H5 goes, at this stage, no. At this stage, trade regulations based, as I understand it, trade regulations based on H5 are regulated by compartmentalization more than anything else, meaning defining what markets you're allowed to vaccinate. So, for example, the USDA received approval in order to vaccinate the California condors because they were a segmented market unrelated to the poultry industry or the beef industry or the dairy industry. But all of those negotiations are ultimately, I think it's safe to say that that's the primary concern now in terms of usage. I don't think availability of vaccine is as much of a concern anymore as the potential effect on trade and making sure that those trade regulations are actually met.
Jen,
my opinion too.
are you seeing another question?
Yep, I saw another question in here. Is Medgene also developing a vaccine for H5N1 in poultry? And is there an adjuvant used in the vaccines?
So yes, all killed vaccine products need an adjuvant. That's the nature of life. And so, yes, we do. We have several adjuvants. We have one for companion animals that we have that's in our products. We have one that's used primarily in swine and poultry, and we have another one that we use primarily in our ruminants. And so what I presented today for H5 for dairy cattle is all the ruminants. We have done trials with poultry and with other species for H5N1. Some of those you may see information on in the near future, but the USDA has yet to see and approve that data, so I'm a little hesitant to talk about it.
If we have folks who still have questions, I'd encourage you to go ahead and type them into the chat box while you're typing. You know, one of the things that we asked early on was if we're feeding this milk to calves, can they become infected? So my question is, can we vaccinate these newborn calves in a protocol that would give them protection at least till they're weaned?
I'm going to answer that two ways. I have to be careful. Our current safety studies have been done on cattle down to 3 days of age. So from a safety standpoint, yes. Now, in terms of vaccination strategies, that all depends on when they're liable to get infected and how they're getting affected. So obviously, if they're getting colostrum, then the colostrum contains the virus. That's going to be a problem. Can't vaccinate after the virus has already gotten there. That's not how it works. In terms of blocking infection or potentially gaining antibody from the milk, that would be a possibility as well. So getting the antibody levels up in colostrum and milk would probably be your strongest route of protection. Yes, you could vaccinate them. They'll get a decent immune response. It takes at least 14 to 21 days before you get a decent antibody response, and the protective is after the second dose at 28 days. So depends on when they get infected.
Jen, are you seeing another question out there?
I'm not seeing any other questions unless I missed anything. People can retype it, or I think we're getting down on numbers here. People can unmute as well.
Yep. Well, I'm not seeing anything come in, so we'll go ahead and do the closing here. Thank you, Dr. Young. Very informative, a lot of information. Fortunately, you have your contact information on screen, so I would suspect you may be getting a few emails with questions. So folks who attended today, we appreciate your patience. It was a large group, and we worked to get your questions answered. Say thank you to my cohort, Jennifer Bentley. I appreciate your help in making this happen. We'll look forward to seeing folks if they're interested, coming to the next program. The dairy webinar will be the 29th, and we have Dr. Cole, who's going to talk to us about the inbreeding issues in the dairy Holstein breed. So thank you again, Dr. Young. We appreciate it, and we will close out at this point.
Thank you.